just to sort of keep you on track as to where we are in the semester, we've had now 3 exams. We have one more coming in just under 3 weeks, and that will cover the last 4 chapters of the course. We're going to have a couple of review sessions that I'll conduct to give you the broad overview sweep of the course. And then the final exam is the first day of finals week. So we have exams a week apart. I apologize for that. I don't get to pick the exam schedule. But it will be a cumulative final. So you might want to start thinking about reviewing past work in the course now, just to sort of anticipate that, since it's at the beginning of finals week and not later on. I can tell you now that roughly half, somewhere around half, 20 to 30 of the 50 questions will be taken from our exams. So using your past hour exams for this semester will be helpful to you. Other questions will be comprehensive across the semester, not necessarily from the exams. Do you have any kind of course-related questions before we start talking about DNA today? Okay. Well, so on the end stretch. Okay. So we're really changing our focus now. We've come to an end of metabolism, properly speaking, with the end of Chapter 16, the integration of metabolism. And what we're shifting into now is a different kind of thinking. And it gets called a number of different things. You could think of it as the management of genetic information, you know, what you have and how you use it. Sometimes it's called macromolecular metabolism because it's making and destroying really big molecules like DNA and RNA and proteins. Sometimes it's called molecular biology. Of course, all metabolites are molecules, so that's kind of a self-serving name. And we can break this down into 3 areas. We can think about the ability to continue information, make it go on existing and persist in those who live after it. And that would be replication of DNA. Replication of DNA and its repair. That DNA is one of the very few molecules that is important enough to cells to fix it rather than just throw it away when it breaks. The second dimension of this is amplifying the information. That is, we've got these genes, this DNA, and we want to use this piece of information a lot, and we amplify it by making RNA molecules from it, and that process is called transcription. So we'll talk about that. Now, some RNA molecules are useful in their own right, but in terms of a variety of molecules, most RNAs have to be translated into proteins. They're messages that encode a protein. So the bulk by mass of RNA is in the ribosomes. The bulk by variety is in the messenger RNA and needs to be made into protein. So we'll talk about translation as our last topic. We're going to begin today talking about Chapter 17, which is going to address the DNA in the upside of it, the fresh side of it. We're going to be talking about topology, its ability to be twisted and shaped. We're going to talk about replication and about packaging, how you can fit DNA into your cells. The packaging problem can be demonstrated this way. You notice from my thumb to my nose, there's this much DNA in each and every one of your cells. So the packing problem is a very important and significant one. All right. So let's think a little bit about replication, the copying of DNA to make new molecules of identical DNA. You remember back quite a ways to when we were talking about the structure of molecules and the idea that most DNA is found as a double helix with base pairs between the 2 strands in what are called Watson-Crick base pairs, where A can pair to T and G can pair to C. And those are complementary to each other. And it was, as soon as the structure of DNA was understood, that it was a double helix with complementary sequences between the 2 strands, it was presumed that probably replication would exploit that complementarity somehow. And then the question became, was this replication process a conservative one or a semi-conservative one? And what that means is, if replication were conservative, all of the old strands would end up together, paired to each other, and the new ones would be over here as a fresh new molecule. A semi-conservative process would be one where in the new molecules, one strand of the double helix was old and the other strand was new. And over here, one strand would be old and one would be new. And you might wonder, I guess that shows up well enough. You might wonder how you could get at that. And there's a profound experiment that was conducted by Michelson and Stahl. And they took advantage of the fact that many atoms come in several slightly different forms, isotopes, that differ in their atomic weight. And so they grew cells in nitrogen-15. So this is the heavy form of nitrogen. And the parental DNA then had nitrogen-15 distributed throughout the strands. When they shifted the cells for replication, for cell growth and to copy over, they shifted it to a slightly lighter form of nitrogen, nitrogen-14. And then they could isolate the strands of DNA and ask, are we seeing patterns that are consistent with one or the other? And the predictions then would have been like this. If it was conservative replication, you'd have some molecules that were all heavy and some molecules that were all light. That would be a conservative replication mechanism. In a semi-conservative one, all the molecules would be intermediate in weight between the heavy and the light. And in fact, what the results showed us was that it was a semi-conservative process with one strand, every old strand having a new strand attached to it, the product of that replication. And this is important because it's going to affect what models and mechanisms of replication are possible. A further dimension of DNA as a molecule that makes it a challenge to replicate it is its topology. Now, topology is that branch of geometric properties that are aspects of shape and arrangement without actually cutting and changing things. So in topology, a teacup and a donut are the same thing. They both have a single hole through them. And one aspect that makes DNA a challenge to replicate is that in cells, in a living organism, most of the DNA is going to be supercoiled. That is, it's not just going to be a helix as a molecule, but that helix is going to be twisted around on itself. And you've probably seen this if you've ever seen a old-fashioned wall phone that has the telephone cord tangled. I don't know if... Is there anybody here who has never had a corded phone? Good. So this is not yet a dead metaphor. The other place you may have seen supercoiling is if you've ever had a model airplane with a rubber band. So if you twist the rubber band and it gets super and tighter and tighter, those are supercoils in the rubber band. And hopefully that metaphor will never die. But you can see these physically. This is an electron micrograph of DNA, and they're showing you how this piece of DNA, so this is a circular piece of DNA from a bacterium, presumably, a plasmid from one. And you can see how it's twisted over and around itself. So the molecule is more coiled than just the intrinsic nature of DNA double helix would require. And this is a result of actively twisting it, more twisty, or untwisting it, over or underwinding it. The fact that most DNA is supercoiled in cells contributes to stabilizing DNA for a variety of reasons. And it means that whenever we are going to deal with DNA as a substrate, so for replication when we're going to copy it, for transcription when we're going to make RNA copies off of it, recombination we're going to see is when you actually cut and paste to rearrange the DNA. In all those conditions, the supercoiling makes a difference. Just a reminder to you that the genes in eukaryotes are linear pieces of DNA. In prokaryotes, they're circular molecules of DNA. But in both cases, they are supercoiled. And in eukaryotes, maintaining supercoiling with linear DNA obviously invokes additional structures to make that possible. People who want to talk about the structure of DNA in more detail can talk topologically about twist and writhe. And twist has to do with the number of helical turns, writhe with the number of times that a strand crosses over itself. And interestingly, you can convert writhe and twist into each other. This is showing you that topologically, writhe and twist are in terms of a topologist, the same thing, they're just organized slightly differently. One of the reasons that supercoiling is good is that it makes DNA take up a lot less space. Again, if you think about that rubber band airplane, the rubber band could get into a lot smaller space when it was supercoiled. The linking number is the sum of the twist and the rive. So you can talk about a particular plasmid of DNA whose properties will differ with its linking number because it has more or less supercoiling in it. So in order to replicate DNA, in order to make new copies, we're going to have to separate those two strands. And when we look at DNA inside of a living cell, we often find that it's naturally negatively supercoiled, so that when you pull it apart to replicate it, that actually helps to relieve some of that supercoiling. If the DNA were not negatively supercoiled, what you'd see would look like something like this. Here's some unsupercoiled or relaxed DNA, and now when you pull these strands apart, you're pulling coils into it as you pull the strands apart. So the negative supercoiling facilitates opening up the double helix for access to the machinery. The topology of DNA is so important that there are actually enzymes that the cells have to allow the cell to change the topology of DNA in directed, appropriate, regulated ways. And because you're changing topology, they're called topoisomerases. So a topoisomer is an isomer. It's the same molecule that differs topologically. And topoisomerases change supercoiling. So they're going to cut the DNA somehow. They're going to then rearrange the structure appropriately and then reseal it. And these are some of the kinds of reactions that topoisomerases can catalyze, depending on which topoisomerase you're talking about. This is an example of a supercoiled circular DNA. This is a relaxed supercoil. They only differ topologically. This would be one of the things that a topoisomerase can do. Actions of topoisomerases can actually result in knotting, an actual knot in the molecule. Or if you have, for example, two circular DNA molecules here, these are independent molecules, but they're caught inside of each other like the links in a chain. That's concatenated. And a topoisomer can unlink them, so they're now two separate molecules of DNA. So these are all examples of topoisomerase changes. And this kind of activity is going to be needed for replication, and it's also going to be needed for transcription. And for once, the nomenclature has some rationale to it, that topoisomerases 1 will cut one strand to accomplish this, and topoisomerases 2 will cut both strands in order to accomplish this. So for once, enzyme names that are really straightforward. So if we look at topoisomerase 1, here we have the intact DNA strand. There'll be a strand break on one of the two DNA strands, so that the other backbone is intact, but this one has been broken in the phosphodiester backbone. It allows this helix to untwist, and when the supercoiling is relaxed, the rest of the molecule will swivel as needed. If you think about the molecule, the phosphodiester backbone is rotationally free to twist when it's a single strand. When it's in the double helix, it's not. And so when it's nicked there, the intact backbone opposite it is free to twist and swivel to relax the supercoiling. If you then seal up that break, you now have a slightly more relaxed molecule. You'll notice there are more turns here than here. So this is a more relaxed form compared to that. In human beings, there's a topoisomerase 1 is a small monomeric protein, and it actually kind of encircles the DNA molecule that it's going to change the topology on. The binding site of interaction between the protein and the DNA itself has many positive charges, which makes a lot of sense because the phosphate backbone of the DNA has a lot of negative charges. And it's actually going to make a covalent bond to the DNA backbone where it cuts it. So it's not going to just let it flop and fly free. There's a tyrosine at the active site. The alcohol of the tyrosine is the place in which you create a phosphodiester bond to preserve, like holding on to the strand of that you've made a nick, and the opposite strand is then free to rotate to relax the supercoiling. This actually makes clinical differences. There's a drug called camptothecin, a very useful anti-cancer drug. And camptothecin is an inhibitor of topoisomerase 1. And so by binding to topoisomerase 1, it will lengthen the period of time that the DNA is in the nicked configuration and not an intact, completely covalent backbone. And we're going to see as we discuss replication and transcription that a nick in DNA is not, in general, a good thing. And so what this does is that camptothecin has the strongest effect on rapidly dividing cells. Since rapidly dividing cells are the ones that are replicating the most and they're transcribing the most, something that interferes with replication and transcription will principally target rapidly dividing cells. Now, there are some other rapidly dividing cells in our body, and hair and gut come to mind, and that's one of the reasons why drugs like camptothecin have side effects that are unfortunate. You can kind of see how that looks like it might well mimic part of the structure of DNA. Now, I mentioned that there's also a topoisomerase 2, and it cuts both strands of DNA. It follows a similar logic in that it has an active site tyrosine, but it has two of them, so each one can bind on and hold on to it. And so if we have here a supercoiled piece of DNA, we can cut it and bring the entire thing through and close it back up again. The topoisomerase 2, unlike topo 1, requires the presence of ATP, and then the ends are resealed to complete the molecule, and so forth. There are, just as there are inhibitors of topo 1, that camptothecin inhibits topoisomerase 1, there's an inhibitor of topoisomerase 2 that you may have heard of, ciprofloxacin, or Cipro. Do any of you remember the anthrax attacks back in 2001? This was an antibiotic that was used against the anthrax. DNA gyrase is a type 2 topoisomerase in bacteria, and ciprofloxacin will inhibit the activity of that. Now, because it consumes ATP, it's actually capable of adding supercoils, so it can increase the coiling of DNA. And conveniently, since ciprofloxacin is targeted against a bacterial topo 2, it does not damage human cells in the process. By preventing the proper processing of the DNA form, you're going to have more cleavage of DNA, or the cleaved things will persist longer, you'll reduce the stealing of it, and in the end up killing the cell. Now, when you think about the challenge of trying to deal with DNA, again, remember that each of your cells has this much DNA in it, actual length. Clearly, you're going to have to organize the process of copying that if you want it not to simply end up as a tangled pile of spaghetti that's knotted together like a pile of old shoelaces or something. And what we've discovered in fairly recent years is that, in fact, replication in eukaryotic nuclei is very organized into what have sometimes been called factories. And what this picture is here is a fluorescence micrograph showing you how the replication centers are organized in a eukaryotic nucleus. You can see some places where nothing's happening because those are places where ribosomal RNAs are being made. But that the nucleus as a whole has many small foci that are engaged in replication. And so these are places at which the proteins are organized for replication, and then the DNA is going to get reeled through to be copied. If in a bacterial cell there might be one or two or very few sites like this, in a eukaryotic cell, 100, 150 different locations for this. So since there's so many locations, there are many forks at which the DNA is being replicated. This is clearly a very useful adaptation for having a lot more DNA. We have roughly something like 1,000 times as much DNA as E. coli does. To give you a little bit of a sense for the relative speed of all these things, if we look at the E. coli genome, we've got roughly, 5 times 10 to the 6th, or 5 million base pairs. The machinery there can replicate 1,000 bases a second. And typically it takes 40 minutes to make a new copy of DNA for E. coli. We have roughly 3 billion base pairs. And our replication machinery is a lot slower. We do about 50 bases a second, so that's a lot slower per base. And if we did this at a single site, every cell would take a month to replicate. You would still be very small, right? But in fact, a human cell frequently can finish replication in an hour because multiple replication sites are engaged in copying simultaneously just on different parts of the DNA. And this is, I think, showing you some of the origins of replication on human chromosomes. Okay. So replication happens at what we call replication forks. That is, parental DNA has been opened up, and the process of replication is occurring at this location where the strand is newly opened. And it's generating new strands that are complementary in the semi-conservative way on each of the template strands. So we're going to look at how it is that the replication machinery can generate new molecules on those templates. Okay. One of the things we're going to see is that all of these macromolecular processes can be thought of as having 3 stages or 3 steps. You have to start it, you have to elongate for a while, and you have to stop it. And I'll try to allude to this each time we come to a new process. But it makes sense that this would be true. You have to have some way to get things going. There's the sustenance or the elongation phase, and then ending it. And for initiation, we're going to have to form the replication fork. We're going to have to assemble all the different bits of machinery that's going to be needed for that. And that only is going to happen at a DNA structure that we call an origin of replication. Extension or elongation is going to be the ongoing, sustained process of making the complementary DNA. And we're going to discover that it's a little different at the 2 strands. It's easy to imagine it just sort of zooming off like that, but it turns out that the structure of DNA doesn't let us do that. So the leading strand and the lagging strand will be a little bit different. An enzyme called DNA polymerase is obviously going to be key to this, but it needs a lot of help to get that all to work. So if we look at this replication fork, we're going to see that we're going to need a number of different components before we can actually make the whole thing go. We're going to need helicases to allow for unwinding. We're going to need single-stranded binding protein because it wants to snap back. We're going to need the DNA polymerase, which is actually going to catalyze the reaction. We're going to see that there's a sliding clamp that helps with this process. A primase, because polymerase only adds to things that are there. A telomerase, which is has its own funny job, and we'll talk about that. And a ligase that will help kind of clean up things at the end. So we have a number of different people to meet and to understand. So the job of helicases is to separate those DNA strands in order to allow access to those bases. So this is a kind of a topoisomerase. And these are going to use the energy from ATP to catalyze that. There's some evidence that they rotate, and they can unwind up to 750 nucleotides a second. So these are fairly potent, structurally modifying enzymes. Pulling it apart is not enough. They really want to snap back together. So the next thing that you can think of conceptually is single-strand binding protein, or SSB. If those strands you pulled apart did snap back, you'd be back where you were. But single-strand binding protein, interestingly enough, binds to single-stranded DNA. Another logical name. And it has a lysine-rich structure. You've got all these phosphate backbone, a lot of negative charge. Lysine has a lot of positive charge. It's a pretty non-specific interaction. It just recognizes the fact that it's a single-stranded molecule. In eukaryotes, the single-stranded binding protein is called replication protein A. And it does more than just be involved in replication. We're going to see that many times if you're trying to do something with DNA, trying to keep it single-stranded can be helpful, and that replication protein A also functions in things like repair and recombination. Okay. Now, the actual synthesis of new DNA is accomplished by proteins called DNA polymerases. So again, a rational name. And their job is going to be to incorporate deoxynucleotide triphosphates. So if you have a molecule of DNA in which part of it is double-stranded and you want to complete the rest of the strand, what the polymerase can accomplish is to take, this is going to be the template strand here, this is the free end, this deoxynucleotide triphosphate is going to be added there, and now you've got a base pair you didn't have before. It's a consequence of the base pairing between the old strand, the template strand, and the incoming nucleotide. It's going to release pyrophosphate, so it's a moderately expensive process. And this growing chain that is the site of addition for the new incoming nucleotide is called the primer. DNA polymerases need a primer to add to. They don't add to empty air. They add to something that's already there. This is a constraint on DNA replication compared to some other things. If we look at this in just a little more detail, here is the existing DNA strand. Here is the free end of the primer strand, the new growing strand. And the 3' hydroxyl is the one that's free and floating in the breeze. And so the incoming nucleotide is going to add from the 5' carbon, going to create a phosphodiester bond and create a new free 3' bond. So we speak of DNA polymerases as synthesizing from the 5' to the 3' direction. That is, the 5' end of the primer is the oldest part of the molecule. The 3' end of the molecule is the newest end of the molecule. DNA polymerases are processive. And by processive, we mean it's an enzyme that does something and does it and does it and does it and does it, does it again and again and again, repeatedly over an extended template. We talked back in the cytoskeleton about dynein being processive. I think we may have talked about some of the glycogen phosphorylases being processive. The DNA polymerases are really processive. And so in any given event, there are going to be multiple nucleotides added into the new growing chain, as many as 5,000 polymerization events in a given cycle of being bound to the template. Now, some of that processivity is due to the polymerase itself, and some of the processivity is due to a protein complex called a sliding clamp. And this is sort of a schematic representation of sliding clamps, how they encircle the duplex and provide a sort of a locus for the DNA polymerase to do its job and keep everybody kind of all together in the same neighborhood. These are a couple of different sliding clamp complexes. This one is a dimer of trimers. This one is a trimer of dimers. But you can see in both cases that there's a six-fold symmetry around them. The DNA polymerase itself has an overall structure that we're going to see again when we look at RNA polymerases, and it sometimes is called a right-hand structure. So if you look at my right hand, and you imagine the thumb and the fingers and the palm is where the DNA is going to be held, And the fingers and the thumb can grab the incoming nucleotide triphosphates to facilitate addition to that template. And you can see the fingers and thumb here with the sort of flat bed in the middle for the DNA template to hold on. There are many DNA polymerases. I don't believe there's any cell known that has a single DNA polymerase. E. coli has itself at least 5 DNA polymerases, and we have as many as 15. The reason there are so many different polymerases is that they're doing their job in slightly different settings. There's bulk replication, where you need to copy the lagging strand and the leading strand. There are DNA polymerases inside the mitochondria, inside the chloroplast, because both the mitochondria and the chloroplast have some DNA of their own, and that needs to be replicated. There are DNA polymerases that play a role in holding sister chromatids together during meiosis. There are other DNA polymerases that play a role in fixing up damaged DNA, and there are almost certainly other functions still that we don't even know what question to ask. So let me just see. I guess that copies up pretty well. This particular picture, you can still see the kind of thumb and fingers and palm overall structure. Their general structure is very similar to each other. This one is one that's involved in translesion synthesis, that when there's a damaged bit of DNA, this is the polymerase that plays a role at copying through that. DNA polymerases are remarkably faithful in how they make their copies. You can imagine that if you don't do it right, there's problems, and we're going to see in Chapter 18 just exactly how bad some of that stuff can be. And so they are, we speak of polymerases as, most polymerases as having high fidelity, that the error rate is fairly small. And there are a couple of ways in which the fidelity is kept high. One is structural, that the active site at a DNA polymerase has a very tight fit between the template DNA and the incoming nucleotide triphosphate that's going to bind there. There just isn't a lot of wobble room. If you don't have a good match there, it's going to be hard to fit in that space. But the second thing is that there is an exonuclease activity that can proofread. So the polymerase is adding bases, and the exonuclease can actually eat them off. This is an example of exonuclease activity. Here we have a template strand. We're going along from 5' to 3', but a base was put in that was not correct. It's a mistake. There's a 3' to 5' exonuclease. So it starts at the 3' end of the growing chain and eats back however far the errors are. So it can proofread for the DNA polymerase. The estimate in typical humans is 1 mistake in every 1,000,000 nucleotides incorporated. That sounds really good. You have 1 billion base pairs in the genome of your cells. So each time cells are copied, there's 1,000 mistakes made. We're going to see some of how those get fixed. This might be a good time to mention that fidelity or accuracy is actually a selectable property, that you can make DNA polymerases be more or less accurate. And when they experiment in this way, for instance, on E. coli, they find they can make the DNA polymerase more faithful, more correct, as well as obviously sloppier. It's easy to think you might make it sloppier, but you can make it more accurate. But in the cells in which the DNA polymerase is more accurate, the growth rate goes down. That the cost of the greater accuracy is taking more time and more energy at each base pair. So there's an optimal level of errors in DNA synthesis. So there are some challenges for DNA polymerase, and one is that DNA polymerase only adds on to an existing 3' end. It has to have that free 3' hydroxyl, so it cannot start DNA synthesis. If you give it a template strand like this and DNA polymerase, it'll just sit there and say, what do you expect me to do? So there has to be something there beforehand. So another enzyme called primase, again, a logical name. Molecular biologists, being unconstrained by structure, get to name their things logically. And what the primase does is it lays down a small stretch of RNA It's an RNA primer. So during DNA replication, there is a period in the process where there's a small amount of DNA-RNA hybrid because the primase has laid down a stretch, not a huge stretch, a short stretch of RNA molecule, maybe 60 nucleotides long, for example. But it has a free 3' hydroxyl, and so the DNA polymerase can extend from that and elongate it into a new chain. This RNA is going to have to get removed and replaced with DNA later on. It's not left behind. So at some point, that's going to get cleaned out. Another problem is that the DNA polymerase only synthesizes 5' to 3'. It has only one direction that it can elongate. DNA, on the other hand, of course, is anti-parallel. It has strands in opposite directions. And so you need some way to copy both strands. Especially since the replication machinery is sort of held still, you have to cope to some extent with the fact that these things are trying to go in opposite directions. And so we talk about leading and lagging strand synthesis, and they're going to turn out to be slightly different. This is a slightly intricate picture, so take your time feeling like you've made sense out of it. Here is the old DNA, and here is the replication fork. And we want to make copies of the template, so we have copies on both strands. There are going to be two molecules of DNA polymerase. You can see the little green circles are the DNA polymerases. The leading strand is going to be synthesized continuously. And so the strand on the left here is being elongated and elongated. And you can see it's very long, and it's all one new molecule of DNA. The opposite strand has to still be synthesized 5' to 3', but it's ending up bubbling out a bit of the template because it's going in, quote, the opposite direction or whatever. And so it does it in kind of a peculiar way. The first time I heard the description of this, I thought, that can't be how it's done. But it was. Sometimes life is stranger than we can imagine. And so here you see a growing chain here. It has its own little primer there. It's being elongated and sort of pushing out the template as it gets elongated. This new piece of primer is going to loop out again. And we're going to end up on this side of having several shorter pieces, each of which have their own little bit of RNA, and they're not linked together yet topologically. So the leading strand is a single continuous effort, just this wonderfully processive molecule going along. The lagging strand is the result of several bouts of priming, elongating, and so on. And this is showing you that in just a little more detail here. Here you have your template strand, a primer, and a primer. These short pieces of DNA and RNA covalently attached are called Okazaki fragments. Okazaki was a brilliant young Japanese scientist, and I think it's worth remembering his name because he was a child during the bombs on Hiroshima, and one of the reasons he died young was because he was a child at Hiroshima. So there's a certain poignancy about his having discovered this mechanism of synthesizing things in pieces. So here we have some Okazaki fragments. This DNA polymerase is elongating in this direction. This one is as complete as it's going to be. We can't leave the RNA there. We don't want RNA in our final molecule. There's a special RNase that can degrade the RNA there called RNase H for a hybrid. It only degrades RNA that's in an RNA-DNA hybrid. So RNase H is a 5' to 3' exonuclease, eating it up. The DNA polymerase continues to elongate, and under optimal conditions, you end up with two pieces of DNA with just a space between them now, with a discontinuous backbone. And that discontinuous place, that little nick in the backbone, can then be sealed by DNA ligase. DNA ligase consumes ATP or NAD depending on the species that we're talking about. It can happen that the DNA polymerase is barreling along, and it gets to the DNA-RNA hybrid area before the exonuclease is done, in which case it might push off kind of like a flap of RNA because it's synthesizing as it goes. So this is a little flap of RNA, and there's a special endonuclease called flap endonuclease that can identify the junction internally. So the other one was an exonuclease eating from the ends down. This is an endonuclease cutting right in the middle. But it still leaves a gap, and the DNase ligase can then close that nick. So if we have an overview of DNA replication, this is a sort of a roadmap for you showing you the different components all on the same picture. Here now the leading strand is on the right-hand side and the lagging strand on the left. So here's your old DNA, the parental molecule, the topoisomerases that are going to be working on it to open it up. Here's a helicase that's actually opening it at that spot, single-stranded binding protein, keeping it open until the polymerases can be active there. This DNA polymerase has made all of these nucleotides from the 5' to the 3' direction. This on the lagging strand has multiple copies of an RNA primase making a little primer, a DNA polymerase adding at the end of that primer, and ligase sealing the breaks after those are all done. If you were to look at molecules of circular DNA, so a plasmid, for example, in a bacterial cell, you actually can see figures like this where in the earliest possible stage there's a tiny bubble, and as replication proceeds, that bubble gets larger and larger as the strands are replicated. You can see how the elongation is making its way further and further down into the molecule. Now, prokaryotes are fine. They've got circular DNA. The polymerase goes around, and it's done when it's done. Ligase can close up that last singular nick. Eukaryotes, on the other hand, have a much more significant problem. Eukaryotes have linear DNA molecules in them, and this is going to be a problem at the ends of the molecules. So let's take a look at why that's a problem. So here we have a template strand, and the DNA polymerase can read out to the 5' end of that template just fine, and then be done, and there you have a new strand. However, at the 3' end of the template, we have a little bit of a problem. If we have a primer here that we use to start off the whole process of polymerizing it, and we remove the primer, there's no place to come in and make the DNA that needs to fill in there. So it leaves us with a 3' overhang that cannot be filled in by the DNA polymerase. So it's a single-stranded DNA overhang that, given the way polymerases work, we can't replicate it in the normal way. Now, single-stranded DNA tends to get attacked by nucleases. So if we were to have two linear chromosomes up here that have just been replicated and they have their hanging free 3' ends, nucleases would come in, they would clean up that extra DNA, and you'd end up with two new shorter molecules of DNA. And at every round of replication, you'd lose a little more from each end, or from one end of the molecule again and again. And so this would not be a good thing. Overall, you'd end up just eating your chromosomes up. So telomeres are structures at the ends of chromosomes that are specifically there to address this problem of the 3' overhang. And telomere just means end, telo for end, like teleology, if you happen to know that word. And then mere is the generic one for body or mass. So the way that the telomeres work is that they have repeats of a fairly trivial sequence. They're using here the example of TTAG-GG. So this is one particular telomere sequence. And at the ends of chromosomes, there are multiple copies of those sequences. And They don't code for anything interesting. They're just there structurally to create the telomere. And if you have a 3' overhang like this at the end of a round of replication, the enzyme telomerase recognizes that and elongates the free end of the molecule, giving the DNA polymerase a little more room to copy out. And then if there's a little free end cut off at the next round, telomerase can re-elongate it a second time. So the free 3' overhang problem only means that telomere DNA is being cut off, not informational DNA. And the telomerase can actually generate new telomere from scratch. It's a kind of an interesting enzyme. It has a small piece of RNA on it that has the sequence complementary to what the telomere repeat is going to be. So it carries inside of itself the template that's needed. And in humans, the telomeres can be anywhere from 5 to 15,000 bases long. Okay. So I've mentioned this, that there's an RNA component to telomerase. There's also a lot of protein to it. And we're going to discover that in the nucleus there are a number of ribonucleoproteins, which are a mixture of RNA and protein. And the enzyme activity is therefore, strictly speaking, a reverse transcriptase. It's taking an RNA sequence and generating a DNA sequence from it. There's some thought that the reverse transcriptase of telomerase may have been the source of the reverse transcriptase in HIV. Okay. So in the development of the body and changes over time and so forth, the activity of the telomerase can change in the lifetime of an organism. So that, for instance, in a 2-year-old or a 5-year-old, the telomerase will be more active, and then in someone who's 60 or 70, they may be less active. Certainly when we culture cells in culture, we find that at some point the telomerase enzyme becomes inactive, and as a consequence, the chromosomes can shorten, and there comes a point when they no longer have the full complement of information to sustain replication and the full range of activities of the cell. Cancer cells typically have an active telomerase, and so they can keep replacing them, and no number of divisions will end up killing a cancer cell. Typical numbers for human cells may be 50 to 60 rounds of division. There are some cancer cells that have been going through cell division since the 1950s. If you've ever worked with HeLa cells were obtained from a woman who has been dead now for many, many years, and those cells go on dividing quite indefinitely. And this was from one of my TAs earlier. Find the key to immortality tomorrow. I got a mop. So maybe that's a good place to break. I'll see you tomorrow on Friday.